which means it's very thin and it's got a moist surface. The list shows different substances in the body. So write the name of the correct substance. So I'm going to use either oxygen, carbon dioxide, or food. So the lungs transfer oxygen to the bloodstream. Small intestine transfers food to the bloodstream. And the mother, she transfers food and oxygen to the fetus. That's the little baby inside the mother. Which part of the cell controls the substances, which substances can enter? That's the cell membrane. The job of the cell membrane, allowing things in and out, depending on size usually. What is the name of the process that allows the transfer of stuff? It's going to be diffusion, unless it's the transfer of water, therefore it's osmosis. Explain how digested food molecules transfer through the mother. So again, for 3 marks, write it down. By diffusion, which means they move from an area of high concentration to an area of low concentration. 3 marks. Next question. It says, snails live in seawater and can take salt. So this is a higher question. We need to know about active transport. The process of taking something from a low concentration to an area of high concentration is called active transport. So this is the opposite to diffusion. And sometimes organisms need to do this. Plants do it often. If there's more nutrients inside the plant cell than outside, they will still use a little bit of their energy and take those nutrients in because those nutrients are vital. And it also happens in marine biology where you've got that salt water, but obviously organisms need fresh water to live, and this is what they'll use, active transport. The chemicals needed, this is a naughty question for 2 marks. Chemicals, you're thinking, oh my god, but really they should say, they've said chemicals because glucose, they could have said molecules to make it easier. They need oxygen and they need glucose because it requires energy, and energy comes from respiration. So anytime you're using energy, you're gonna need oxygen and glucose, and you're gonna get rid of carbon dioxide, and you're gonna get rid of water. Use a diagram showing some things. I'm sure you can read that if you need to pause the video. It tells which cell, A or B, will take oxygen in the fastest. I said B because there's a greater concentration difference. There's a lower concentration of oxygen already in you, and it looks to me that the outside of this cell and the outside of that cell has the same concentration of oxygen. Therefore, I think the quicker rate will be, well, I don't think, I know the quicker rate will be in this one, because this one contains more oxygen, so it's got the greatest concentration difference. So they've got less molecules inside the cell, which will diffuse into the cell. The diagram shows a plant cell from the root. What is the name of the process caused when the water moves into the cell? It's called osmosis. Okay, it's where water moves from a high concentration to a low concentration through that semi-permeable or partially permeable membrane. What is meant by the term partially permeable or semi-permeable? It allows some substances, some, not all, some substances or molecules through. These cells have been placed. These cells have been placed into a concentrated solution. Finish sentences about the cell. Cells have lost water, therefore the cell contains and not pushing against the wall. Okay, so this one, I'm not even sure where to go through this one because you won't use. We've not learned this word. It's not in your spec, and I don't even know where to go there. But we'll do it anyway. The cell wall, and so their cell wall pressure has dropped. Therefore, the cells become flaccid, and then the cell membranes come away from the cell walls. This is plasmolysis, really. We don't need to know it. I don't think it's higher. Let's not go there. Just conscious of time because I got a lesson in 5 minutes. Let's zoom in on that. The world's first power station, which just uses water, has been built in Norway. The station has 2 large tanks. Blah blah blah. So let's have a look what it's asking us. It says, by what process does the water transfer from one tank to the other? Well, you can see there's a partial permeable membrane there. It says that one has got seawater, and one's got pure water. So it's going to be via osmosis, because it's the movement of water. Which tank contains seawater, and give a reason? Well, this tank B contains seawater because if we had the same amount of water each side, there's less water in tank B because it's got more salt. So that salt takes up space. So if there was the same volume, this is going to be more concentrated. This is going to be more dilute at this side. So the water, because there's a higher concentration of water this side, it's going to pass through this semi-permeable membrane, and it's going to have equal amounts of water on each side. But obviously this side has got salt as well, so it makes the volume increase. So this one's got water and salt. This one's just got water. Yeah. So that even though that could be they could be equal concentrations now of water either side, because this one's got salt as well, it pushes that volume up. The movement of water will be towards a more concentrated solution. The concentrated solution is in tank B. Explain how water is transferred from one tank to the other. Well, I've said water transfers from a dilute solution, so an area of weak concentration, a high concentration there of water, tank A, to a more concentrated solution. Concentrated meaning that it's got salt in across a semi-permeable membrane. Okay, quite a tricky question there. What is an enzyme? An enzyme is a biological catalyst. What's a catalyst? A catalyst is a protein. It speeds up reactions without getting used up itself. So it doesn't get used up, so it can do its job over and over. What is the name given to the structure represented in diagram B? Well, when the substrate actually joins to the active site, or the enzyme, the active site of the enzyme. So the white is an enzyme, the black is the substrate. We call it an enzyme substrate complex for 1 mark. Explain why the denatured protease, D, this one, is unable to catalyze the separation of the 2 amino acids. Well, you can see there's a specific shape on these amino acids, and because it's been denatured, the active site shape has changed, so therefore it can no longer fit. So the active site of the enzyme has changed shape. State 2 factors that affect the rate of enzyme-controlled reactions. Well, I've put 4 down. pH is a massive indicator of the rate of these enzymes. The temperature as well, so usually the warmer it is, so long as it's not too warm, the faster the reaction will be. I've said the concentration of enzymes, so how many enzymes are in there, and also the concentration of the substrates, so how many substrates are available in that area. In the diagram, the carbohydrate starch is broken down into sugar. So we're getting starch. Starch is a large molecule, and it breaks down into a small molecule called glucose. This begins in the mouth and uses the enzyme amylase. The graph shows the rate of reaction. So describe the rate. So whenever you get a question like this, it's usually worth 2 or 3 marks. You're looking for the trend. So as the rate of reaction, so as the rate of breakdown, increases, okay, as it increases from 2 to 7 pH, it increases so it also increases. So as the rate of breakdown increases, so does the, let me rephrase that. So the rate of breakdown increases from, and I've not been able to take a photo of that, that's it. So from the pH of 2 to 7, where 7 is neutral, the pH, as the pH increases there, so does the rate of breakdown. But then after the pH of 7, when it gets more alkali, it starts to decrease again. So then decreases from pH 7 to 12. The optimum pH of this, and I've drawn a line to that peak, is 7. Very conscious of time now, and this is my last page, so I'll do this very quickly. We've got a 20-minute video. Amylase is an enzyme which is found in saliva. It only digests starchy foods. Identify the optimum temperature. I've identified it there to be 37 degrees. Describe why amylase stops at 42. It's becoming denatured because it's a high temperature. The substrate no longer fits the active site, and therefore the enzyme changes its shape. Describe the shape of the graph so describe the trend really. The amylase activity increases from 0 to 37 and then decreases rapidly, so it's denatured. Explain why amylase only breaks down starch. Well, the shape of the active site is specific to these starch molecules, and that's why usually each one enzyme will only do one sort of substance. And then for the last question, some amylase is found in the mouth. You can see there, if you did read the question, that it needs to produce, so amylase is produced in the mouth. It gets denatured in the stomach, therefore the small intestine needs to make some more. So I've said the low pH in the stomach will denature the amylase, therefore they need to make more in the small intestine. And that's that video.